And our last speaker of this segment is Peter Hunter of the University of Auckland. He's a distinguished professor of engineering science and director of the Auckland Bioengineering Institute. He's renowned for his work in computational physiology and computational biology, and in using engineering, mathematical, physics, and chemical process concepts in modeling biological systems. His talk today is about the 3D scaffolds in SPARC. All yours, Peter.
Okay, thanks very much, and hi everyone from Auckland. So Bernard, I think, has already talked about the background to the NIH SPARC project, so I don't need to talk about that. And what I was asked to talk about was particularly the scaffold, the common coordinate scaffolds that we're developing for a number of the organs in the SPARC project. I'll talk about just two of those. One is the heart scaffold and one the stomach scaffold. And what we've done is to develop a sort of biological CAD platform. This is all open source, freely available software on GitHub. And what it allows us to do is to create particular organ anatomical features of organs that we can tailor with Python scripts to suit the topology of any particular species. And then these allow us to define, to whatever level of resolution we want, the anatomical features that we want to capture and annotate. And it means that we have a framework where we can define all sorts of tissue properties relative to that material framework. And as I'll show in a minute, these coordinate systems are material coordinate systems, so they move as the organ moves, the coordinates move with it. And those coordinates are defined relative to an external fixed system. So then we can export meshes for simulation at whatever level of refinement is required. And these meshes can be made, the scaffolds, the generic scaffolds can incorporate tissue structure or vasculature, for example, or particularly for the SPARC project, the intracardiac neurons. And then we can generate these for particular species. And there are some topological differences, particularly in the number of pulmonary veins, for example, that enter the left atrium varies between species. And of course, we can also capture individual variability, as I'll show for the stomach in a minute. So one of the ways that we use these scaffolds is in the context of what we call the Physiome project, where we're dealing with the physics of different processes, in this case, coupled electromechanics, where we've got a knowledge and measurements of tissue structure that we can incorporate into these scaffold frameworks, but we can also embed a lot of cellular properties that are then used as part of the simulation of, say, electrophysiology on the heart or mechanics or other aspects. So that's shown in a bit more detail here, where we go from molecular scale to cellular scale to tissue structure. It's the fibrous structure of the heart, to the whole heart, the physics of electromechanics on the whole heart. And then we use these models in clinical applications for fitting to patient data for helping with diagnostics or surgical planning. So this is just one example where, just to show the deformation of the mesh as the heart is beating. And what you're looking at here is the subendocardial surface with one scaffold element shown here, and then we've just texture mapped onto the back of that element the tissue structure. And these arrows here are the eigenvectors of the strain tensor being computed during the deformation, just to show particularly the shearing behavior that goes on with a lot of these tissues. So I'll just go through the stomach, which is another organ, obviously, that undergoes fairly large deformation. And so it's the same process of defining a generic scaffold and being able to fit it to individual species. And there is a reasonable amount of variation across species, particularly where the esophagus joins the stomach. Rats have a very big forestomach, for example. And down on the right, you can see the material coordinates, both around the stomach, but also through the wall as they vary, and we can capture the local fiber orientation. And this shows some of the cell type regions. This is data from the John Finesse group at Melbourne, and also the vasculature that we can then incorporate into these stomach models. And this is showing the changing fiber orientations as you move across the wall of the stomach. And then this one here is just as part of the SPARC project, showing some of the way that we deal with data from the Terry Powley group at Purdue who are imaging the stomach in the rat. And the way they do it is to make cuts along the lesser and greater curvature of the stomach and open it out. And often their measurements are in relation to that opened out stomach. So what we got them to do was to do micro CT imaging of the stomach that we can then fit our scaffolds to, segment, and then fit the scaffold to this particular data set as shown here. And that means we can then open these scaffolds out in the same way that the experimentalists do. So you can cut them along the greater curvature, for example, and then this looks like the preparation that they use experimentally, and we're now mapping their data of the neurite pathways into that cut tissue and then closing it up again. And then because we want to get good averages across a number of stomachs, this shows 11 micro CT images where we're fitting the scaffold to each one individually, but then doing an average. And we can do principal component analysis to then look at the dominant modes and be able to reconstruct any one of these from a combination of modes. And then we can take that average. This is just showing an exploded view of it to show the different layers that are important for the the autonomic system. And then just displaying some of the traced neural pathways, just a few of the neural pathways traced onto this average stomach. And I'll finish there, and just this, I've only shown a tiny bit of what we're doing for the SPARC project, but these are the people involved in Auckland in the project, including Bernard, who you've heard from, and a lot of the scaffold development work is being done by Richard Christie, and a lot of the stomach work that I showed was being done by Kumar. So thanks very much.
Any questions for Peter? I'll start with one. Peter, those two organs are organs that have very large contiguous lumens.
Yeah.
The heart and the stomach. Any thoughts on how to deal with an organ that doesn't have a large contiguous lumen? Liver, the kidney, the brain, where there may be sub-elements that are repetitive, they may not be repetitive. Any thoughts on how to handle that kind of an organ?
Yes, what we do is to have a hierarchy of material coordinate systems. So, I mean, I've mainly focused here on the material system that defines the geometry, the anatomy of the organ at a macro level. But in the lungs, for example, we'll have detailed models of the airways going through the multiple generations of the airways and the vasculature. And those structures are also modeled, but they're embedded in the host coordinates of the organ. And of course, the organ is embedded in the whole body coordinate system. But that can go on indefinitely. So we can embed a variety of material coordinates depending on the spatial scale of interest that always allow you to move from that scale up to the host scale above. So we have done models of the kidney where we've got the calyx defined and the vasculature defined, and that's all within the context of the lobes.
Great.
Peter, very impressive work. Thanks so much for presenting. I was wondering how many different individual mice have you used for the stomach, and how many other individuals have you used for the heart?
it was actually a rat that I was showing rather than the mouse, but for that one, the average there was based on only 11 actual different rat experiments. But what we're doing at the moment, or what the Terry Parley group is doing, and we're fitting the scaffolds to, is for a number of rats, all of the ones I showed were at a fixed state of inflation, fixed filling of the stomach. But he's now doing a number where he varies under controlled conditions the level of filling of the stomach. So we're then able to capture the scaffolds at different levels of distension to understand the way the stomach inflates when it's distended. At the moment, the total numbers are still only, I mean, I think the plan with the neurites for the stomach are about probably 300 rats are being processed for those neurites, because they can only do a certain number of measurements of neurites on a given preparation. So they do quite a large number of preparations. So part of what we're doing is setting up automated pipelines to be able to process the data to create the scaffolds automatically, and the neurite locations within those scaffolds. For the hearts, we've done a whole variety of species. I mean, we've done about, I think, 27 dog hearts. We've done a lot of pig hearts. We've got a database of human heart anatomy based primarily on MRI. So the numbers are in the tens rather than the hundreds on the whole.
Do you have a guesstimate for how many human hearts it might take to really capture a human heart across ages, across sex, across ethnicity, all adult human and healthy?
I think it depends on the level of resolution you want. I mean, what we find with the heart is that the fiber orientation fields, the variation of fibers that occur when you go transmurally, is very consistent across species. And there's a good reason for that, which we understand, I think. It's just that the fiber organization is very much reflecting the loads that the wall is carrying in different positions in the wall. The sheet structure is also reasonably consistent within one species, but varies a bit more across species. And the sheet structure is very much determined the way that the tissue shears, and that each species has a slightly different mechanical performance in the way that the tissue shears. So they have slightly different tissue structure for that. The vasculature is reasonably consistent. I mean, there's a number of typical morphologies, but it's reasonably consistent. The Purkinje fibers, on the other hand, are extremely varied. We haven't really found a lot of consistency in Purkinjes. And of course, we've yet to determine for the autonomic nerves, we don't know yet. That's the data that's coming through from SPARC. So I'd have to say it just varies a lot depending on which anatomical structure you're talking about.
That is very...
In terms of aging, I'm not sure we really have a good feel for that yet, the variation across ages. That's yet to be done.
This is Zarina. First of all, I want to thank you. It looks like you stayed up all night. I don't know who came up with this idea.
The early morning.
Keep you awake.
7 hours.
I've been a long time admirer of your work. So again, wonderful stuff. So I want to get your opinion, since we did keep you up all night. What do you think about building these sort of scaffolds from inside out as opposed from outside in? So I was interested in the vasculature, so you could build around the vasculature, like if you were going to do a three-dimensional printing of any organ, just the way it develops around the vasculature. So what were your thoughts about that? As opposed to try to capture the shape outside and then try to go for changes in shape. The vasculature will modify with the heartbeat or with the breathing of the lungs or the expansion of the stomach, everything.
Yeah, so remember that when we define any of those internal structures like vasculature or airways or neural paths, they're all defined in material coordinates, so they all move with the deforming host tissue. So the vasculature, sorry, I should have shown you an example of that, but the vasculature does deform as the heart deforms through the cardiac cycle.
Yeah, and the relationships between the cells around the vasculature remain similar. So they all slightly shift, but they stay together, sorry.
Absolutely, yes, yes. And I mean, I think in any of these problems, you need to be able to link molecular right through to whole organ, whole body physiology. And so it's really important that the frameworks allow this hierarchy of ways you define information at different scales in a consistent way that allows for the deformation and the physics of the processes going on at that scale, particular scale.
Thank you.
Sam, did you have a question?
Yeah, I've got a quick question. So, you know, and you just sort of touched on this, Peter, about making sure that we can go through all these different scales. And one of the things that we've talked about with CODI was that we wanted to make sure that we had these nice landmarks so that we can figure out, oh, hey, this is how the tissue that we take here lines up with some sort of CCF. Do you have good models for how these landmarks change as you inflate these organs, or how the landmarks would otherwise modify, through, for example, with the heart during the regular interaction of them beating, for example?
Yes, I mean, the vasculature, of course, always provides very good landmarks because the bifurcations of the vessels are...
Yes, but not all of the landmarks that we're using are necessarily vasculature, though.
No, sure. Yeah, so every organ is different, and every organ will have its own particular set of anatomical structures that become good landmarks. And it's important to capture those in a material coordinate system and to annotate them appropriately so they can be found through searching. But generally, we find it's not too difficult to find appropriate anatomical landmarks in any given organ.
But that's finding the landmarks. I'm saying, how do the landmarks change? Because if I'm trying to say, oh, look, I see this, but then it looks different, either a pathologist or maybe machine learning won't be able to correctly identify it.
Okay. Yeah, I mean, obviously the landmarks, as I was saying before, follow the deformation of the tissue. So that's not an issue. But sure, there will be different imaging techniques will be presenting those landmarks in different ways to the viewer. That's true. So if you're trying to automate the detection by machine learning of those landmarks, I don't know the answer to that. How difficult it would be to always be able to reliably capture particular landmarks with a whole variety of different automated imaging techniques. Yeah, that's a good question, and I don't think we've done enough.
I think that some of your questions are not very clear to us, and I think it's usually a problem that occurs when different specialties are talking to each other. So for instance, you ask a few questions also on the challenge. You asked this question. I'm not sure what you mean about landmarks changing. You assume they are changing, or are you asking they are changing, or what do you mean?
So I'm assuming that the landmarks to some extent change.
What way?
So at least spatially. So, you know, if you have the heart, for example, it's beating, correct?
Yeah.
And so, you know, if I'm saying, you know, look, I'm trying to look at some spatial asterism I see in the tissue, and, you know, I see one configuration here, and then, you know, the configuration
of the tissue does not change.
But if the configuration does not change, then the tissue has to be absolutely static.
No. So what changes is there are slight variations in the size or potential distances between various elements, but the configuration, the patterns don't actually change.
So can I, sorry to interrupt here. I've actually got to go because I've got a call with NIH, our regular Friday morning call.
Oh my God.
Apologies for that. But I'm very, very happy to come back online at a later stage if that's useful.
That would be great. Thank you.
Thank you, Peter.
Okay, cheers.